Okay, so let's have the class. And what we will talk about today is, first of all, we want to see the results of the spread and pore plating. So first of all, when you look at your plates, let's give an example. Let's say this plate. And you will see 10 to the minus 6, there's a colony there. Then 10 to the minus 5, you will see there is actually lots of colony there. And 10 to the minus 3, you can even see lots of colony. It's a cluster there. So when we record this, we usually say 10 to the 3, 10 to the 5, and 10 to the 6. We will say these two are T and TC, two numerable to count. Now 10 to the 6, when we look at that, you will see this is approximately the number is between 30 to 300. Therefore, we would love to count it. So who want to count this? Sean, can you go ahead to count this to see the number?
Yes.
Okay? That's very important. So Sean will count it. And then this is spread plating. And the pore plating is right here. Now when you do the pore plating, you will see basically the bacteria is actually everywhere. So we'll see another 3 plates right here. This is 10 to the minus 3. There's lots of bacteria there. 10 to the minus 5, also lots of bacteria. And 10 to the minus 6, you see there's a colony there. It's very clear. And you do see there's also those tiny dots. So we're not going to be count right here. Okay, so Sean, you can go ahead to count these plates as just a representative of the whole class. So we'll see. So again here what we see is 10 to the 3, 10 to the 5, 10 to the 6. These two are T and TC. So when you're counting, I will suggest you to use a marker to label it every time you count it. Okay? So let's see the number. For pore plating, you only need to count the large one.
Oh, okay.
Yeah, only count the large one.
There's like about 60.
Okay, then this is pore plating is 60. So what is the final population? That equals 60 multiplied by 10 to the 6 CFU per ml. That is equals 6 multiplied by 10 to the 7 CFU per ml. Now most of the time for microorganisms, we change them to the log 10. 6 multiplied by 10 to the 7 CFU per ml. So what is the results? It's approximately 7.7 log 10 CFU per ml. Okay? Now this is, you should know. This equals 10x equals 6 multiplied by 10 to the 7. So this x we can transfer into the log 10. Because bacteria is too large, So population is huge, so we usually transfer them to the log. So Sean, what is the number?
284 CFU.
How many? 284. Okay, very close. So 284, what is the final number here? Their final number is 284 multiplied by 10 to the 6 CFU per ml. Again, we're going to be transfer them to the 10, log 10, 284 multiplied by 10 to the 8 CFU per ml. So that is almost equals 8.3 log 10 CFU per ml. Okay, here you will see a result, which is the number of the spread plating is larger than whole plating. Did you see it? Why? Because the mild heat of melted agar will killing bacteria, could kill bacteria. Okay, so that's the reason. So that's what we did for pulp plating and spread plating. And this will be going through the whole semester for everything. So we will be just do it again. So just let you have an idea, okay? Okay, we're going to do today's work. Today we will do is recognize bacterial media and the corresponding to the foodborne pathogens and do street plating. Okay, what we're gonna do today, so one more time, you will have your plates there, that is E. coli. We will have Staphylococcus aureus right here. This is Salmonella, and unfortunately we do not have the Listeria here, so we will skip the Listeria, we'll do it next time. Okay, so we have Staphylococcus aureus, E. coli, and the Salmonella. So we're using the selective media to talk about that. So first of all, I want to talk about some of the concepts. Number one is called selective media. Okay, selective media, which means there is a certain ingredient in a bacterial media which is favor certain bacterial growth and inhibit others. And we will do some of them today. I'll give you some of the examples. We have a Macanthi agar. This is specifically selective for E. coli. We have Magnetosalt Argument. This is selective for Staphylococcus aureus. And we will do XLT4 agar, which is specifically for Salmonella. Okay, so we want to introduce you all of these, but before I introduce all of these, you also want to have another concept we want to let you know. It's called the differentiator media. Differentiator media, which means there is ingredients in this media where can differentiate the bacteria from certain ingredients in medium will generate biochemical reaction which can differentiate the bacteria based on the color. And I want to mention this because McConkey agar and Magnetosalt agar, this is also is a differentiated media. So that's why I want to mention that concept too. Now we want to talk about these one by one, so we want to talk. First of all, McConkey agar, it is a purple, looks like this, a purple color. Now why it is a purple? Because the major ingredients is crystal violet. Because of the crystal violet, so only gram-negative bacteria will grow. And gram-positive bacteria will be killed. The reason is crystal violet will attach the peptidic glycan. So gram-positive bacteria is killed. The reason is crystal violet attack peptidoglycan of cell wall. Okay? Now I want to also mention McConkey agar, this is also a differentiated media. Why? Because of lactose. Because of the lactose, they could be differentiated, become lactose fermentation positive and the lactose fermentation negative. If lactose fermentation positive, this is pink color, which is pink colony. Lactose fermentation negative is colorless. Why? Because inside of the McConkey agar, there is a pH indicator called meso red. At the lower pH, when the lactose fermentation generates acid, it will show yellow color. Oh, sorry, it will show pink color. Okay, so this is something I want to mention. Then, second one is Magnetosalt agar. Magnetosalt agar for selective Staphylococcus aureus. This is also a selective media because of salt. Salt will be inhibit bacterial growth because unbalance of osmotic. If you're in a salt solution, the bacteria will be burst, absorb too much salt. So in this agar, approximately they may have 3 to 5% of the salt. Lots of the other bacteria will be killed. Staphylococcus aureus, we also call it a halophile. It will be survived. Now, the thing that I want to also, this is also a differentiated media because of mannitol. Mannitol is a sugar. So mannitol could become mannitol fermentation positive and also mannitol fermentation negative. If mannitol fermentation positive, it is yellow color. If mannitol fermentation negative, it is colorless. Because same thing, there is a pH indicator called pheno red inside. And this pheno red at acid environment will be showing yellow color. Okay, so just let you know. Now the last one, what I want to mention is XLT for agar, for salmonella. What does X means? Xerose, lactose. T is tergetophor. Okay? XLT for agar, I just want to make sure what I'm doing is correct. Yeah, lactose, lactose. The selective ingredients of this XLT4 agar is digital form. It is a very strong selective ingredient. Any injury of the cell, of the salmonella cell, will not be grown there. Now, salmonella grown XLT4 agar is actually a black colony. Or some of the textbook will say red colony, black center. The reason is there is ingredients in there which is metallic ingredients. And the metallic ingredients in there, it will be going through anaerobic fermentation, will let sulfide oxidize to sulfide acids. Therefore, it will show you black colony. However, this black color will be faded if you put the agar on the room temperature for a long time. The reason is the oxygen will be reacted, it will be reduced back. Okay, so there's something I wanna mention before I move on. Yeah. Okay, the reason is very correct. Okay. So these are the things we will do practice today. Now we want to measure another agar, which is a blood agar. So first of all, you need to know blood agar is an enriched medium because they containing 5% sheep blood. So, just 5% sheep blood, they find it's very similar to the human blood back in 1970. And higher nutrition of hemoglobin, so the bacteria will be grow dramatically. But I also tell you this is a differentiated media. Why? Because based on whether you hydrolysis or we say lysis blood cell, it can be differentiated from alpha hemolytic, beta hemolytic. So it's a lysis, not really hydrolysis, gamma hemolytic. Alpha hemolytic, usually it's a light green color, incomplete lysis. Beta hemolytic, it is transparent because it is complete lysis of blood cell. And gamma hemolytic is actually nothing happens. Okay, and you should find Staphylococcus aureus is beta hemolytic. Okay, so we gave you an introduction of all we have. Now what are we going to do today? It's very simple. So I want to be drawing right here. We will do a very simple streak plating, and we will talk about these again next week, Tuesday. So the first thing we're going to do is streak plating. So what we're going to do, E. coli onto Macanthi agar. Staphylococcus aureus, Magnetosalt Argument. And Salmonella XLT4 Agar. Okay, how to do streak plating? This is the Agar plates. Of course, you're going to have to do label. Label your name, bench number, and your bacteria. Now, you either use loop solutions or the Agar plates. So you're going to use your loop to pick the column. And you do very intensive first cut streak using loop. Then you have to flame it. Do a second cut, less intensive. Then you flame it again, do the third cut, and then flame it again, do the last one. Every time you have to come in from the original places. So this is we call it's a serial dilution on the solid agar. And after incubation, you should see there's a colony grow there. And we really want those because that represent a pure culture. What is a pure culture is a group of cells coming from one mother cell. That is what we need. Especially in the food and microbiology area, we do the isolation and the identification. The second work, what I want you to do is we want to see the differentiate it impacts on the blood agar. So what we do is the blood agar, we will be split them onto 3 parts. And we will write SA indicate Staphylococcus aureus. SL indicate Salmonella. And the EC indicate E. coli. And you use your loop to do a very light streak. And we will do the incubation. We are trying to see the different hemolytic status. So that's very simple. Now don't forget also label at the back of your agar. Okay? So this is a very simple way to do. And I'm going to show you how to do it. Just use one example. So Sean, you're going to help me a little bit. And I'll just use Macanchi agar. Just come here. And I want to have gloves. Do you have gloves? Thank you very much. So we're going to show you how to do this. You can do all of this by yourself. But sorry, I missed the Listeria mouse pathogenes, but we will be mentioning it again. Okay. So first of all, you're going to label. This is Macanchi agar. You want to label CS, EC, and the bench number. Let's say number 1. Okay. Now let's have a plate. Just have one of the plates, and we have a hockey stick. You want to turn it on. Okay, now you want to use a loop. This need to make sure they must streak. Okay, so first of all, make sure it's very streak, and it has to be burned to red, and then burn a little bit on the top. Okay, this is sterilize the process, a little bit of anger there. Okay, now we're going to pick a bacteria from it. So we pick a bacteria. We call down on the side, so we pick bacteria. Okay, how we do it? Very intensive at the beginning. You will do a very intensive first cut, like that. Then you're going to have to burn it. That's very important because you're going to execute it, and then from the first cut, decide to do a second one. Call down. You see the marker there? So do a second light streak. Now you have to burn it again. Okay, burn it again. Hold on. Turn around from the second cord, do a Third one. Then you burn it again. Hold on. From the side of the third one, do a last one. Okay? You can see, clearly can see there is a marker there. And then you do the incubation. We hope to see the colony there. Okay? So this is the one I want to show you. Now the second one, what I want to show you is blood agar. So blood agar is like this. You will see at the back there's a blood agar. So what you do, of course you're going to label. Okay, ECS number 1. We want to label it into 3 parts. Okay, we're going to label SA, Staphylococcus aureus, SAL, and EC. So label like that. Now what we do? We will be using the loop. Using the loop again. Okay.
Oh, we moved left. I thought this was the right one.
Sorry. So hold on. Now for Staphylococcus aureus, this one you have to shake a little bit. Okay. So we pick some from there, from the liquid. And then Staphylococcus aureus, we just do a very gentle. That's it. Now let's say E. coli. We do the same thing. We're gonna pick from the plates. So pick from here. And we do very gentle. Speak like that. Okay, the last one for Salmonella, what we do? So coming from here. Okay. Hold on. Get the solution. Then we will do on the other side, so this side. So it should be looks like that. And then we will do the incubation. You will see the growth status of these. Okay, we can do a separate one for the exterior later on when we move on to the hot dog. Okay, so that's all we have.